Hello, my name is Madison Pachulis, and this video is about how to perform 16S rRNA PCR. PCR stands for polymerase chain reaction and is a method used to make copies of specific DNA segments. PCR amplifies the DNA sequences to generate millions of copies of that particular segment from a small portion of the DNA. For this video, we are focusing on 16S rRNA PCR and the bacteria E. coli. There are 5 ingredients to a PCR tube. First, the template DNA that you want amplified. In this case, we use genomic and colony E. coli. Next, we need primers. Primers are single strands of DNA that are complementary to DNA sequences on either side of the region of interest. We used a forward and reverse primer for each PCR tube. The standard primer, in this case 27F, attaches to the start codon of the template DNA, and the reverse primer, in this case 907R or 1522R, attaches to the stop codon of the complementary strand of DNA. We also need a PCR master mix, which contains the Taq polymerase, dNTPs, and buffer solution. Last, we add H2O to correct the concentrations of the tubes to prevent degradation of the DNA. If done correctly, each tube should have a total of 50 microliters. Each tube will contain different combinations of DNA and primers so that each one can be individually visualized by gel electrophoresis. Tube 1 contains genomic DNA with a concentration of 10 to 50 nanograms per microliter. Genomic DNA is chromosomal DNA from the bacteria. This tube also contains the forward primer 27F, the reverse primer 1522R, the master mix, and H2O. Tube 2 contains DNA of E. coli. Colony DNA is used to directly amplify specific regions of vector DNA from the bacteria. This was obtained by taking one single colony from the petri dish and adding it to the tube. This tube also contains the forward primer 27F, the reverse primer 1522R, the master mix, and H2O. Tube 3 contains genomic DNA with the forward primer 27F, the reverse primer 907R, the master mix, and H2O. The purpose of 2 different reverse primers is because they have different sequences that will attach to and help amplify different regions of the DNA. Tube 4 contains colony DNA, the forward primer 27F, the reverse primer 907R, the master mix, and H2O. Tube 5 contains a no template control in order to monitor DNA contamination and use as a negative control. This tube also contains the forward primer 27F, the reverse primer 1522R, the master mix, and H2O. Tube 6 contains a no template control, the forward primer, the reverse primer 907R, the master mix, and H2O. The PCR procedure involves 3 important steps: the denaturation process, the annealing process, and the extension process. Denaturation heats the double-stranded DNA to separate into single strands and take place between 55 degrees Celsius and 95 degrees Celsius. There are multiple cycles in order to produce a large number of copies. The annealing process takes place at around 55 degrees Celsius. This step allows the DNA primers to attach to their complementary sequences on the template DNA. The extension process takes place at 72 degrees Celsius. This process extends the primers and allows the new strand of DNA to be made by the Taq polymerase enzyme. The tubes are refrigerated and stored until needed. Following PCR, gel electrophoresis can be used to visualize the fragments and separate and analyze the fragments of DNA based on their size and charge. Thank you for watching my video on how to perform PCR in 16S rRNA of E. coli. For further information of the processes of PCR, you can watch Canal Divulgacion's video on the polymerase chain reaction.